And which bioinformatic tool or software you have used?
Bioinformatics software, ma'am, like BLAST. I can use BLAST tool to find out the mirror repeats of these genes.
No, why you have selected that gene? Why you have selected?
Ma'am, because these genes are used in heart regeneration, that's why I can take all important gene which is published in NCBI.
कौन है? उत्तम मैडम है ना?
Okay.
Yes, ma'am.
Okay, the next one is P002. Dhankar Mamta Yadav Dipti.
Hello, ma'am. Am I audible, ma'am?
Yes,
Yes.
Good afternoon, everyone. My name is Mamta, and I'm a research scholar at Baba Mastanath University, Rohtak, Haryana. And my research topic is identification of mirror repeats in CAD4 apoptotic gene of C. elegans. So what is apoptosis? As we know that when a cell decides to kill itself, and our aim is to find out mirror repeat in CAD4 apoptotic gene. So various genome size of the C. elegans is very small. It consists of various type of genes.
Madam, who is presenting? Let them open their video, no?
Yes. Participants, please note that during presentation, please on your video.
Okay.
So genome size of C. elegans is very small and it is distributed over 6 chromosomes. Different types of repeats like tandem repeats, direct repeats, palindromic sequences, transposable elements are present within the genome of C. elegans, but one type of repeat, that is mirror repeat, is rarely studied in genome of C. elegans. So what is mirror repeat? Mirror repeat is a repeating sequence that possesses a center of symmetry, and our aim is to find out mirror repeat within the gene CAD4 gene. This gene consists 2,847 base pair and it have 8 exons. And this is the location of gene on the chromosome number 3. So we use a simple bioinformatic tools to find out the mirror repeats within all exon of the CAD4 gene. And this is the result which shows that the number of mirror repeats at different threshold. And we obtained 10 mirror repeats in exon 1, and 1 mirror repeat in exon 2, 3, 4, and 4 mirror repeats in exon 5, 6, and 7, and 6 mirror repeats in exon 8. So total, we got 31 mirror repeats in the exon of CAD4 gene. So future molecular and future study are also required to find out the exact role of mirror repeats. There might be possibility they play an important role in apoptosis. Like VNTR, it is a part of repetitive sequence that play very important role in human DNA fingerprinting. Thank you.
Madam, what is the difference between, you have told that is satellite DNA, these are also repeat, then this mirror repeats, and what is use of your study? What is difference between that simple sequence repeats, that is mini satellite, and your mirror repeats?
Ma'am, mirror repeat is that repeat which is repeated on the same sequence with reverse order. But other repeat like tandem repeat is that repeated sequence that is repeated from head to tail fashion. It is a category of, or you can say it is a type of repetitive DNA.
Okay, then what is use of your study? If you have studied mirror repeats, if it is more, then what happens? Whether it has any role? It affects that gene coding genes. It affects with that gene.
Ma'am, our preliminary aim of our research is to find out the mirror repeat because no one has studied the mirror repeats. But on other repeat study have been done.
Okay. Okay. Your aim.
And biological. Yes, sir.
Okay, Mamta, your aim and objectives are correct. But what is the outcome of what you are expecting?
Sir, our future aim is to find out to form any primer of these repeat. These repeat play any role in apoptosis or not, or any other process.
Okay,
Okay.
means if the repeat is more, then whether it affect the that gene coding or that you want to study. Okay, okay, okay. And which software or which bioinformatic tool you have used, or only BLAST?
Ma'am, we simply use BLAST tool.
BLAST. Okay.
Yes,
Okay.
ma'am.
Okay.
Okay, thank
Okay.
you, ma'am.
Next. Thank you.
Malvakar madam.
Yes, sir.
Yes,
Now,
sir.
the next one is PZ03, Dr. Nisha Varde. Please start your presentation. Are you there, Dr. Nisha? Yes. Please share your work. Your time has started.
Didn't पाहिजे होती म्हणून मी
Sir, maybe proceed further.
Okay, okay.
Okay, sir. The next one is PZ04, Happy Patel. Please share your work.
three is absent, madam.
She is there, but I think she is not online.
Hello. Good afternoon, everyone. Myself Happy B. Patel. I am master degree student and I am studied at Department of Bioscience, Civil Normal South Gujarat University, Surat, and I am doing right now internship under Rutuja Charitable Trust. My today's topic is premenstrual syndrome in women. What is a premenstrual? Premenstrual is a word made up of by two words, premenstrual and syndrome. Premenstrual is a phase of before 7 to 14 days of menstruation, and syndrome is a set of symptoms or conditions that occur together and suggest presence of certain disease. We know premenstrual is suggests menstrual cycle related something. In menstrual cycle, there is a four phase. First one is menstrual phase, second is follicular, third is ovulatory phase, and fourth is a luteal phase. In luteal phase, we known as some symptoms are occur, and these symptoms are also called as a premenstrual syndrome. In premenstrual syndrome, premenstrual in reproductive age 15 to 40, women are suffer from premenstrual syndrome symptoms. Symptoms are mostly physiological, emotional, and physical symptoms. In mostly 150 to 200 symptoms are found, and mostly found symptoms like headache, breast tenderness, abdominal cramp, weight gain, skin fatigue, skin changes, pimples, fatigue are physiological symptoms, and emotional symptoms mostly found in female premenstrual phase is anxiety, anger, poor concentration, decreased mood, wanting to be alone is a emotional symptom. In menstrual cycle, changes in hormonal level, estrogen and progesterone is responsible for premenstrual symptoms. Mostly, main reason behind premenstrual is not found yet, but estrogen and progesterone level up and down is responsible for premenstrual, and some family history of depression is also responsible for premenstrual syndrome. Diagnosis. In premenstrual syndrome, there is no medical test or no any particular medicine for premenstrual syndrome. but we can treat and we can prevent premenstrual syndrome through some treatment like physical activity, meditation, nutrition
Just
management.
यनकंची सर म्यूट करा म्यूट यनकंची सर फक्त
I'm
आवाज
sorry.
कुठून येतोय काही कळत नाहीये.
There is no particular medicine and no particular medical test for diagnosis of premenstrual syndrome, but we can prevent premenstruals through some treatment like physical activities, meditation, hormonal therapy like some hormonal injection are prevent for premenstrual syndrome. In India, there is not much research about menstrual condition and menstrual disorder, but awareness of related premenstrual and premenstrual related issue is extremely low in India. PMS is experienced by more than 85% women world over. PMS is complex but highly treatable, and some symptoms are in some female symptoms are very severe, and this form is also called as a PMDD, is a premenstrual dysphoric disorder. And this one is my reference. These 3 books are related to premenstrual syndrome is my reference. Thank you. Thank you for listening me peacefully.
Madam, it is your review paper, yes?
Yes, ma'am. I am doing MSc right now.
Yes. So how many research paper you have studied for writing this review?
5 to 6 research paper I am studied, and some books.
Madam, by studying only 5 to 6 research paper, whether anybody can write the review?
No, ma'am, not only. I'm read a review article, research paper, and some books related to premenstrual, menstrual, and after this I am doing review on premenstrual syndrome.
Okay, so what is your conclusion?
Conclusion is that in India, more than 85% women are affected premenstrual syndrome symptoms, but in India, awareness and research are very extremely low about menstrual disorder and premenstrual.
Okay. Okay.
Thank you very much, ma'am.
Patel, according to you, what you are giving the suggestions for these teenagers? Because we have reviewed this one of the very interesting subject. Madam,
Happy Patel, are you
No,
there?
she is mute now. No, she is mute.
Yes, please.
What your suggestions to the any children, and what is the way to you are going to be suggesting these problems, or you are going to be find out any?
Suggestion is that we need to aware every reproductive age girls for about this.
Already in Indian education system, in this age girls, there is a special care is there.
Yes.
Regarding this issue. Because along with that, or instead of that, what you are going to be advising that teenagers?
We need to them educate for about this type of disorder, menstrual disorder.
Okay, already this is there in our education system. Have you seen a present education system?
Not as much as, it's very extremely low in our education system.
What is the difference between research article and review?
Excuse me, sir, I can't understand.
What is the difference between research article and a review? because you are presented to the review.
Research article is actually take a more time, and it's take up for PhD student. I am not right now PhD student. I am just master degree, and I am doing internship. And
No,
throughout
being
this,
being a master degree, how you are writing that you are written your title is the review.
So for that, I have asked her how many research paper you have read regarding that one. So because you are writing the review. Because
Okay,
writing
next.
review, it requires a lot of reading.
yes,
Each and
ma'am,
everything. Yeah.
Madam,
Okay, next.
presently students don't know what is the research paper, what is the review paper,
Yes,
what is the
yes,
publication,
yes, yes,
what is
yes.
the thesis. What,
And
what
for
are they
review,
doing?
yes, for review, how many books, how many years is required for that study? Okay, next.
Next.
thank you.
Okay, ma'am. The next one is P805. K.A. Gazri.
What about the 03?
03 was there,
03?
sir, but not responded. Okay. Yes, you can start.
Hello, good afternoon. This is my poster, but for better viewing, I will like to share PPT, right? Just a minute, yeah. Is my screen visible?
Yes.
Yes.
Okay. So today I'm going to present my work about Tridax procurements of promising analgesic agent. So Tridax procurements, which is most of the analgesic drugs, narcotics or non-narcotics, those are having some kind of side effects, and most of the time they do not cure the root of the pain, and that's why we are trying to find out some plant-based painkiller or analgesic agent, which may be more useful as compared to the presently available painkillers. And Tridax procurements, as we know, that it is a weed, as I have shown you in the picture, that it is a widely available weed, and in case of most of the tribals and most of the folk people, they use this Tridax procurements from time immemorial, as or they simply apply the juice or potis on their wounds or inflamed region, and that's why we have taken this Tridax procurements as our plan to investigate its uh, analgesic action, and according to references, it is also immunomodulatory, and in our study we have collected this Tridax procurements from the garden of Government Vidarbha Institute of Vidarbha, and it is identified according to flora of Maharashtra. And for this study, we are using fresh leaf juice of Tridax procurements, because this is the tradition where the most of the farmers or folk people, they apply the fresh leaf juice, and that's why we are trying to understand the analgesic property of this fresh leaf juice. For our study, we have used mouse as our animal model. So mouse of male and female mouse of age 3 months, we have selected. The weight is around about 25 to 30 grams. And for that, our animal house is also registered.
Madam, only first slide is on screen.
Okay, I will share it, stop share and share it again. Okay.
Yes, yes, yes. Now it is okay.
Are the slides are moving?
Yes, sir. Yes, madam.
Okay, so the plant material is Tridax procurements, as shown in this photograph, and it is a weed which is widely available all over the Asia, and the fresh leaf juice we are using here for understanding its analgesic action. Our animal model is mouse, male and female mouse we have taken of age 3 months, weighing about 25 to 30 grams. Our animal house is registered under CPC SEA. So for inducing pain,
Madam, Gazra madam, in which university you have conducted this experiment?
Sir, Government Vidarbha Institute of Science and Humanities, Amravati.
That registration number is from which university? Animal registration number.
Yeah, it is the registered animal house of Government Institute of Science and Humanities, Amravati.
Amravati. Amravati, because we can't see that number also, because it's going to be merging.
Okay. Sir, shall I read it?
No, madam. Next.
Okay, so for inducing pain, we use formalin. 20 ml of formalin is injected subcutaneously in right hind paw of the mouse. No need to give any anesthesia or so for that. We can handle the mouse in one hand and we can inject it in its right paw. It's a routine method for inducing pain. So we have divided our animals into 3 major groups: control group, which received sterile water; experimental group, which received fresh leaf juice of Tridax procumbens. Here the experimental group is.
Gazra madam.
Ha, sir.
Experimental design is wrong, because for the formaldehyde is using for the inflammation, inducing the inflammation rather than the analgesic.
Sir, it is according to Huskar and Foley.
Madam, it's a standard I'm talking for the analgesic induction of the inducing the analgesic activities or studying the analgesic activity. Different chemical is using formaldehyde or carrageenan used for the inducing the inflammation or inflammatory activity. Anti-inflammatory activity.
We have referred it. Formalin is also used for study of analgesic activity also. Shall I proceed?
Yeah, madam.
Yeah, the experimental group animals, they receive fresh leaf juice of Tridax procumbens. The half of the animals, they receive the treatment 30 minutes prior to pain induction, and half will receive the fresh leaf juice of Tridax procumbens 60 minutes prior to pain induction. The positive control group, which is treated with diclofenac sodium, which is well known non-steroid analgesic, and this group is also divided into two. The diclofenac is received by some animals at 30 minutes prior to pain induction and other at 60 minutes prior to pain induction. Now for observation, we have glass observation chamber which has mirror floor for easy observation. So licking of paw, it has been observed. Licking indicates the induction of pain. So pain is observed in 2 phases: early phase, which start immediately after induction of pain, which lasts 5 to 10 minutes, and it is observed and it is recorded. The licking events are recorded every after 2 minutes. Then late phase, it starts after 15 minutes. So we observe it for 60 to 120 minutes, and these observations are recorded after every 5 minutes. Then we have recorded number of leaking events as well as time spent in leaking, which is measured using stopwatch. And data is analyzed statistically using average, ST, and ANOVA. So these are the results for number of leaking events. For a better viewing, we will see the this graph. So the light colored bars are for the early events and dark colored bars are for the late event, and we can see that the experimental group 1 and 2 as well as positive control group 1 and 2, it is acting. It reduces the pain up to some extent. The results are more significant as far as the experimental group 2 and positive control group 1 is concerned. Then as in late phase, we can see that this experimental group 1 is more effective and it is highly significant as compared to control, as well as compared to positive control group also. The late phase results are more promising, and these are the results related to number of leaking events. And when we observe the time spent in leaking, those are more promising results. We can see that time spent in leaking, it is reduced drastically in early phase. We can see that in experimental group, both early and late phase, the results are very promising, and the time spent in leaking, it is reduced significantly in experimental group 1 and 2,
Madam,
as well as in positive control.
Gazri madam, what is the difference between the stars 1, 2, 3? Because we cannot see that what is the difference between 1 and 2, 3.
What, sir?
Star 1, you have shown star 1, star 2, 3 stars. What is the difference between 3 stars?
These stars, those are for indicating the significance.
No, but you have not written it at.
Madam, what is the level of significance?
Sir, P is less than 0.05 for 1 star. P is less than 0.01 and P is less than 0.001 for 3 stars.
Madam, let me know what is the difference between this 3 different P value.
Sir,
what is the difference between this 3 P value?
Sir, when we study it statistically, we need to define the level of significance, right? And
according to your research, P is equal to 0.001 or okay, it is a 0.05 is not correct for this type of work.
So that's why it is not. We are not saying that it is highly significant results, right? So when the p value is less than 0.001, we are saying.
Next.
मॅडम तुमचा आवाज नाही येत आहे.
आवाज नाही येत मॅडम. आणि अनोवा म्हणता, वेर इज अनोवा? वेर इज अनोवा हियर?
मॅडम लेफ्ट झाल्या बहुतेक. काय हे रेंजच गेली त्यांची.
हॅलो.
हा मॅडम तुमची रेंज गेली. हा आता बोला.
आता
कविता मॅडम, वेर इज अनोवा इन दिस स्टॅट?
Sir, we have also carried out ANOVA, but I have not given the ANOVA tables here.
मॅडम तुमच्या डाटा मध्ये ते अनोवा बसतच नाही. इट्स जस्ट टी टेस्ट बस. Because only 2 things are there. No, one is controlled and another is treated.
Yeah, but we have compared this experimental group 1 with positive control and experimental group 2 with control and positive control.
Madam, time is more.
What is just I want to know what is the name in Marathi? What is this plant?
दगडीपाला म्हणतात त्याला.
But already its effect regarding this analgesic it is known.
No, madam. The anti-inflammatory effects are well studied.
Means previously nobody has studied analgesic. It is not known for the analgesic. This dagdipala, because it is commonly using plant.
It is commonly used from times from a long time. But
yes, and many papers are there or not regarding this analgesic activity.
Regarding analgesic, specifically analgesic activity, I found very scarce papers, and it is mainly used as wound healing agent. Wound healing agent.
And this nimicillate.
पहिलंच गाजरे म्हणून तुम्ही सांगितलं इट इज यूज्ड ॲज अ ट्रेडिशनल मेडिसिन ॲज अ टाइम इमोबरेबल. पहिल्यापासून खेड्यागावमध्ये सगळं वापरतच आहे.
दगडीपाला वापरतात. Yes. And one more, this nimicillate you have given to the rat or that mice, whether it is
diclofenac.
Diclofenac. Okay, diclofenac. But whether previously any scientist has given this diclofenac to mice?
Madam, diclofenac is used as
what is the effect of
analgesic
that?
agent.
No, but whether it is given same effect with mice or something, whether the research has there regarding this diclofenac on mice?
that we have not referred. What we have done for positive
No,
control.
but because it is maybe possibility diclofenac it is using in human being, how the dose you have
Diclofenac is used in all mammals, means for dairy or others also diclofenac is used as painkiller.
yes, but that doctor they have defined the dose for in dairy animals diclofenac for that.
Accordingly we have calculated the dose for mice.
Means you have taken the reference of that
Yeah,
dairy animals.
accordingly we have calculated.
Because in mice I think
according
whether
to the body weight we have calculated it for mice.
Okay, madam. Time is over. More than 5 minutes
okay,
later.
okay, madam. Okay, madam.
So formalin in this pen, as
Okay.
we have seen that
Okay, madam. Okay.
Thank you, Gazre ma'am.
Thank you, madam.
Now the next presentation is P806 Deshmukh RS. Are you there, Deshmukh RS? Okay, we will proceed for the next one. The next one is P807 Purvi Tarsaria. Please share your work. Purvi, your time has started.
Hello.
Yes.
Yes,
you're
ma'am.
audible.
Yes.
Good afternoon to everyone. I am Purvi Tarsaria from Veer Narmad South Gujarat University. I am studying in MSc Bioscience and right now I am an intern in Rutuja Charitable Trust. I am going to present a poster on
Share
the
your
topic a review on
screen. Purvi madam, share your screen.
Please
Yes,
share
yes,
your
sorry, ma'am.
poster. Yes.
Yes, sorry, ma'am.
Okay. Unmute, Purvi. Unmute. माझी स्क्रीन शेअर केली काय तुम्ही तिकडून?
मॅडम सगळ्याकडे तुम्हीच दिसायला लागले मॅडम मार्सामध्ये.
काय झालं काय? मॅडम काय झालं?
Purvi, hello. Purvi, present your poster. Share your poster.
मी काही केलेलं नाही.
दीपाले
sir.
कोणाला झालं ना त्याला बाहेर टाकायचे ते हे व्हायला येता.
Yes, sir.
Who has completed?
Yes, sir.
हा.
Yes. Purvi, unmute yourself and please start.
Yes. Unmute, Purvi, unmute.
मॅडम मी काय म्हणता इथं पोस्ट कडे काय तर त्याचं अनम्यूट झालं का बघा की.
From here, it is okay, sir. Both mic
Right now
and
it is unmute.
video.
हा पूर्वी
त्याला नंतर प्रेझेंट करायला सांगा की.
Okay. Purvi, we will take your presentation after this next one. Till then, please. The next one is PZ08, Radhika Soni.
Yes, ma'am. I am audible.
Yes, yes.
हा yes yes yes.
Okay, ma'am.
Radhika, please switch on your video.
Okay, ma'am. Yes, ma'am.
Start
Ma'am,
start.
as I'm
Okay start. Yes
Okay.
yes yes start.
Yes, ma'am. Myself, Radhika Soni. I am a research scholar in Mohanlal Sukhadia University under the supervision of the Giriman Agda, ma'am. My poster presentation topic is the potentiality of the nanoparticle for the management of treatment of metabolic syndrome. In introduction, metabolic syndrome is a collection of pathologies like diabetes, glucose intolerance, dyslipidemia, insulin resistance, hypertension, obesity, non-alcoholic fatty liver disease, cardiovascular disease, etc. Metabolic syndrome is characterized by the high triglyceride, low HDLC, elevated blood pressure, and the fasting blood glucose. In India, about 10 to 30% people are suffering from the metabolic syndrome, in which abdominal obesity is the one of the major risk factor for the insulin resistance and the type 2 diabetes and cardiovascular disease. In the treatment of the metabolic syndrome, nanomaterials can be used because of their more efficacy and minimum dose requirement. Here I am presenting my on polymeric nanoparticle. Polymeric nanoparticle are based on the polymer within the size range from 1 to 1000 nanometer and can be loaded with the active compound entrapped within or surface absorbed on the polymer core. On the basis of their structure, there may be two types, nanocapsules and nanospheres. Firstly, nanocapsule. In the nanocapsules, a drug is loaded inside the polymeric shell and it released when required. In nanosphere, drug can be absorbed inside or may be absorbed onto the surface of the polymeric core. Examples of the polymer used in the nanoparticle synthesis are the ketosine, alginate, dextran, polylactic acid, and etc., which used in the treatment of hypertension, oxidative stress, inflammation, and glucose resistance and blood glucose control. And here I am using an example of ketosine-based nanoparticle. Ketosine is a promising carbohydrate biopolymer in biomedical field due to its chemical and pharmacokinetics properties. Ketosine show its beneficial activity in protecting and proliferating pancreatic beta cells, lowering hyperglycemic condition, and prevention impaired lipid metabolism associated to metabolic disease. Ketosine is naturally degradable and biocompatible and non-toxic to human body. Here there is in ketosine also used in the regulating the glucose and lipid metabolism in metabolic syndrome. Here I am first diagram is shows about the ketosine used in the glucose homeostasis. Firstly, in the ketosine is elevate the increase the adiponectin and leptin stimulation, and in pancreas it increase the insulin secretion, in intestine it increase the GLP-1 secretion, which help to glucose homeostasis. In muscle cells it increase the GLUT4 expression and translocate this GLUT4 receptors on the plasma membrane. In liver, ketosine nanoparticles decrease the activity of PCK enzymes, which help to maintain the glucose level. And in lipid metabolism, ketosine also help by down regulate the lipogenesis and adipogenesis. Here I am concluding the prevalence of metabolic syndrome due to the urbanization, inactivity, and excessive consumption of energy is increasing, and also a source for many other diseases like diabetes, atherosclerosis, and heart attack. Polymeric nanoparticle increase the function of drug by the keeping them from breaking down and providing constant release. Therefore, polymeric nanoparticles can be beneficial in reducing the amount of used drug and reduce the side effect also in the treatment of metabolic syndrome.
Madam, in this, you have not done any experiment. You have just collected information
Yes,
that ketosine based,
yes.
yes, ketosine based,
Yes.
because it is already present in different papers. They have done the different experimentation also
Yes,
regarding
yes.
that.
Yes,
Yes. So
Yes,
it is
I
just
am
you have collected. You have not
review.
done any.
Yes, ma'am. No, ma'am, no. I'm just
Yeah.
reviewing.
Because this is known fact that nanoparticle,
Yes, ma'am.
they are having more efficiency, and they can be tied with different, but you have taken only one ketosine based.
Yes, ma'am.
And yes, it
Only.
is used in many diseases. Okay.
Yes,
So
ma'am.
scientists,
And
they
it
have
is
proved
more
already. They
efficient
are
than,
using.
yes, ma'am, than metal-based nanoparticles.
Yes.
Yes.
Okay.
Thank you, ma'am.
Next.
Okay, sir. The next is Jadov VD. P809.
Am I audible, ma'am?
Yes.
Yes. Radhika, please stop sharing your screen. Still we are having the same screen that you shared us.
दीपाली कुणाचं झालं ना त्याला डिलीट करा.
Yes, sir.
कोण कोण झालेलं आहे ना त्याला हे करून टाकायचं.
Yes. Next one, PZ09. Jadov. Yes. Start.
Hello, ma'am.
Yes.
Can I start?
Yes.
Good afternoon. My name is Vaishnavi Jadhav. Today here I present my poster on the topic Study of Butterfly Families from Proposed Butterfly Conservation Reserve, Mahadare, Satara. Abstract. The beautiful creature of nature, butterflies, belongs to a kingdom Animalia, phylum Arthropoda, class Insecta, order Lepidoptera. Because having a scaly wings, the order Lepidoptera consists of 6 families of butterfly. All these families are present in the Mahadare. Mahadare is an ecotone between a tropical semi-evergreen and tropical deciduous forest. It is a diverse in a butterfly fauna. The study gives idea about the butterfly families present in the Mahadare. My mentor Sutar Yasar has been studying butterflies at Mahadare for the last 5 years, and we saw the butterflies from all the 6 families there, and we observed their characteristics. First, we see the general anatomy of butterfly. The butterfly body is divided into the 3 parts: head, thorax, and abdomen. Head bears same eye, antenna, proboscis, and palpi. Thorax.
हॅलो स्पीकर.
Thorax. Hello.
मॅडम हे बेसिक काही सांगू नका हो. काय तुमचा आउटकम आहे ते सांगू टाका की काय हे डब्ल्यूडब्ल्यू सगळ्याला माहितीच आहे.
तुम्ही रिसर्च काय केलं किंवा तुम्हाला काय
हो,
सापडलं
सांगा की
तिथं?
आता.
Madam, these photographs are taken
Yes,
by
sir,
you, or it is from internet? Madam, these photographs you have
taken from
taken from
internet.
internet. So
Internet, madam.
madam,
Yes.
so for the research paper, for the presentation, you can't take any photographs from the internet. If you have studied, you are saying that I have reviewed that these many butterflies are there, all the families. So you have not taken the photograph, only you have seen that one. And you are
No,
telling that in
we-
your locality. It is not allowed, madam,
Yes, ma'am.
to take the photographs from internet. And these butterfly I have seen in my conservation area. It is not done.
This presenter is a PhD student or MSc student?
MSc
MSc
student.
MSc student तुम्ही हे सगळं फोटो वगैरे ते ऑनलाईन मध्ये आहे आणि हे बुक मधून आहे तर सगळं ते बोललं हे काय तुमचं काय नवीन थोडं ऑब्झर्व तुम्ही काय केलेलं आहे तर सांगा की बाई.
But
Okay,
how many butterflies you have observed? Which species?
Hello, ma'am.
From
त्यांचं काउंटिंग चालू आहे मॅम, अजून झालं नाही कम्प्लीट.
Okay. Basic question. How you were differentiating the butterflies and moths?
Depending upon their sitting position, butterflies sit flat, and depending upon their sitting position, their antenna, their color, we can differentiate.
Antenna. In butterfly, how many types of antenna are there? In moths, how many types of antennas are there?
No, sir, I am not aware about that.
मग काय तुम्ही केलेलं काय आहे आता? Next.
okay, चला next घ्या.
Next.
Okay, sir. The next one is PZ10 Rakshe SS.
Yes, ma'am.
Yes. Please continue.
मॅडम ऐसा ये काम आहे ना मॅडम काय कामाचं नाही आता. Madam Malevkar, what is the Z06, Z03?
Z06 is not here, sir. Not joined. And 07, she was having problem. Purvi.
नंतर कदाचित करेल ती.
Yes. After this, we will take her.
चला okay.
Kylie, continue.
With Z10.
Good afternoon, everyone. Myself, Kylie. I'm student of Nanoscience and Technology, studying in Mahadar Ecological Institute, Satara. Today, I'm here to present poster on introduction of nanoscience and technology. And here is the abstract. Nanoscience is the study of systems in nanoscale and nanotechnology. Is the ability to systematically organize and manipulate properties and behavior of matter in the atomic and molecular levels. Through nanoscience and nanotechnology, it has become possible to study and create very useful functional devices, materials, and systems on the 1 to 100 nanometer, which is 1 billionth of meter length scale. After that, there are some of the examples in nature which relates to nanoscience. First one is lotus effect. It has super hydrophobic power of self-cleaning. The leaves of the lotus flower exhibit extremely water repellent properties due to micro and nano structure on leaf surface. This structure allows the plant to have magnificent self-cleaning function. Second one is Zico effect. Zico have unique ability to scale vertical surfaces with strong attachment of their toes to the surface coupled with easy and rapid removal. They are able to do this due to attractive forces of million of nanoscale hairs on their toes, known as setae and capitula. Third one is butterfly wings. Butterfly wings are a layer of nanoparticles separated by layers of air. The thickness of layers changes colors that we see. These nanostructures are coated with hydrophobic wax crystals approximately 1 nanometer in diameter. After that application, First one is medical science cancer treatment, textile making antibacterial. Third one is material science flexible materials. Fourth one device glucose sensor. Fifth one is environment conservation. And last one is conclusion. The atomic scale and cutting edge fields of nanoscience and nanotechnology, which are considered to lead us to study nanotechnology industrial revolution, are likely to have revolutionary impact on this study things will be done. Thank you.
Madam, you have just given the introduction of nanoscience and technology. It is already known, it is already there in many books, in many, and researcher are doing research on this. So again, what new thing? Just you have given information to us. Okay, you have collected it something and you have taken the picture from internet and you have shown it to us. Yes,
ma'am. This is only informative. Yes, ma'am.
yes, you have not done anything new.
No.
And
Next,
from
madam.
your work. Okay, madam. Next.
The next one is PZ11. CB Zamdade. Are you there?
Yes.
Yes. Okay. Ramya, your presentation code is PZ12, na?
11, ma'am.
Okay. Continue.
Is this visible, ma'am?
Yes.
Good evening to one and all. I'm Ramya, PhD research scholar from Presidency College, Chennai. I'm working under the guidance of Dr. Uma Maheshwari and Professor Erumalai sir from Madras University. As we all know, this water is a major purpose for the drinking and the recreational and all the activities. I have chosen the lake of Viranam, which is in the Kadalur district, 4 hours of distance from Chennai. This is because why I have chosen the lake is because whenever the Chennai, the metropolitan cities like Chennai facing the drought, we will be receiving the lake, water source from the Viranam lake only. So we wanted to analyze the heavy metal analysis as well as the cyanobacterial growth and the fish mortality. For that, I know this lake itself is 18 kilometer Veeranarayapuram lake. Earlier it was been mentioned after the Chola, Parantaka Chola one. So we have chosen 24 different parameters I have analyzed and concluded in after analyzed with statistical analysis, I found out that biological oxygen demand was there. So I have isolated the cyanobacteria from it in a media of BG11. So here I have serially diluted and tested in agar plate as well as in BG11 broth culture, and initially I found out 40 different of mixed culture. From then I have isolated a single exonic culture was a difficult part, so I've isolated it, and then morphologically identified these oscillatorials and the form medium, and then after I've gone for the 16S RNA sequencing, I found out that this organism is belongs to Deciphilum species, and this one is a form medium species, and yet to be I have submitted this in the NCBI, whereas this one I had to receive 2 times. I have repeated the sequencing and yet to get the report. Then but the morphologically both have confirmed unicellular, exonic culture, and then I have gone for the extraction here using the aqueous methanol extraction. I have done 75% of aqueous methanol for both the samples, that is for Deciphilum and the form medium for the secondary metabolites analyzed, and after that I have gone for the evaporation, and then this Deciphilum formidius were lyophilized and both the samples I have gone for the biomass, and after the biomass I have lyophilized the sample and gone for the ultrasonication. These now I'm doing the ELISA, that is the ADA specific microcystin detection. So that microcystin detection, for that I have nowhere found out that microcystin specific kit in India overall. So I have ordered it from the Eurofin company, which is from USA. It will take 4 months, and now it is in the customs to get cleared. So I'll be receiving it by this week probably. So to analyze the microcystin content of the cyanobacteria. So my aim is to, I have also analyzed given for the LCMS purpose for finding out whether these two organisms specifically is there any microcystin is present or not. I had to receive that from IIT Bombay. And this one I'm planning to do that invertebrate bioassay for the cyanotoxin, and I have got the 60 such species of since everybody is using this Danny fish for this analysis, now it is in endangered, so we are not able to get this particular species in Chennai, that is in Kolathur, where the fish market is a major fish is being distributed throughout the India. There itself I'm not able to get it, and but somehow I've got the few species, a few 40 around. So I'm going to perform this bioassay by next week, and I have extracted again the biomass. The little quantity is there because there is a quantity of the biomass is very less when compared to the plant extracts or the algal extract. The cyanobacterial biomass quantity is less. So I found out one cyanobacteria which has been analyzed using the BLAST 16S RNA. I have got the specific results which is not been matched with the NCBI. So I've got the novelty work and yet to get the patent of it. So only these two which I have got species identification. So I am going to perform with the cell line. Cell line it is under the process, and the MTCC cytotoxicity assay is being carried out, and yet to find out the microcystin. So these are all the bioassay which is being used for the cyanobacterial toxin analysis, especially from the place where the water being used for the drinking purpose. And the results are morphological identification done, and 16 ssRNA sequencing and phylogenetic analysis done for the species, and one submitted for NCBI, and the extraction of cyanobacterial using aqueous methanol extracts have been done, and the toxin assay using the LCMS and ADA specific ELISA kit and using the cell line, and also the is under the progress. Thank you.
Okay, nice, madam. Okay.
Thank you.
Okay.
Thank you so much.
Sir, there is any question?
No.
It's a clear, no doubt.
Yes, exactly. Okay.
Thank you, Ramya. The next one is PZ
Thank you.
12. PZ 12. SG Roger. Are you there? Okay, the next is PZ 14. Sorry, PZ 13. Yes, ma'am. PZ 13. Gaikwad. Yes. Please, yes, please share your presentation and start. Okay, Gaikwad GD, please wait. Purvi is sharing her screen. Okay, Purvi, start.
What is our number? Purvi number?
Yes, sir.
No, no, not
077, sir.
Purvi. Not Purvi has shared, but I think
PZ
13 has shared. 13.
yes.
Preliminary study of ant diversity.
Okay, Gaikwad, then start. Okay, ma'am. After this, we will take presentation from Purvi.
Yes, yes, start now. This one.
Start,
I am
Gaikwad.
Gautami, the student of Rajeshri Chhatrapati Shahu College, Kolhapur. I am presenting the poster on a preliminary study of ant diversity in the college campus area. We select this topic for to find out and collect the variety of breeds and identify the breeds. In our ecosystem, the every part of ecology is very important for the constant environment, where the ant is also part of ecology, which is help to irrigate soil, improve drainage, improve soil chemistry, disperse seeds, prey on pests. In our college campus area, we planted some traps for to know the how many kinds of breeds are we found in some traps. We planted near about 15 to 20 traps with the help of some methods like oil trap, bone trap, chocolate trap, sugar solution trap, detergent trap, etc. traps we planted for a specific time like 1 hour, 2 hours, 3 to 4 hours. After this specific time, the variety of ant species was attracted through the smell, which is depend on ant diet. Then we capture the photos of ants in a live form, which is attracted to the traps and collect them in ethanol for preservation and the study purpose. Then we study the collected ants morphology, like color, shape, size, and some parts of body, which is visible through microscope. And with the help of Google Lens, we find the tentative names, but we're not sure about Google Lens. And then collected samples were also in the preserve form for next level study at ZSI, which is Zoological Survey of India at Pune. For this project, we use some references to make the study of project become easy and clear data. We find the result through study shows that there is a large diversity in ant species. Their their habit also differs from different species to species. Total 20 different species we are collected. Thank you, ma'am.
Hello. Kai now, Kai Malakar Bhai, they are presenting on mobile because we cannot see their name, anything. What is her name?
Gautami
Gautami,
Deluka.
Gautami.
Gautami. Classify the ants. Just to classify the ants.
Yes, sir.
Tell me a classification of the ants.
Ants are from kingdom Animalia, phylum Arthropoda, genus Insecta.
Order, family.
Family from Formicidia, and order is Hymenoptera.
Order is
Hymenoptera.
Hymenoptera.
Yes, sir.
Okay.
Thank you.
Okay, Purvi. Yes, Purvi, share your work. Your time has started. And all participants are requested, if you are presenting your screen from mobile, then hold it in the horizontal way so that we will observe your slide properly.
Madam, what is the number of this Purvi?
PZ 07, sir.
07.
07.
07. Review of, okay.
Hello, am I audible?
Yes, you are audible.
Hello.
Yes, yes,
Hello,
My
you-
start.
screen is
start.
visible?
Yes,
Yes.
screen is visible.
Okay. Okay, thank you, madam. As you know already, I am Purvi Tarsaria, and I'm studying in MSc Bioscience Zoology in Veer Narmad South Gujarat University, Surat, and I'm from Surat, Gujarat. I'm going to present a poster on a review on burnout syndrome in professionals. So I'm start. What is a burnout syndrome? Burnout syndrome is a psychological condition triggered or characterized by exhaustion from work, feelings of cynicism, and detachment from the work is known as burnout syndrome. Now, people are getting confused with burnout syndrome and depression and stress. So there is a minute difference between burnout syndrome and depression. Is that in burnout syndrome is affected people are depressed or detached from their work only, while in depression the entire work, entire day activity, they are getting depressed. So the burnout syndrome is different from depression, and it is necessary to study burnout syndrome because nowadays people are detached from their work. They are unsatisfied with their work and not feeling well while doing jobs, and the performance of many people in their job are getting poor and poor. The major group of the, according to the previous paper, which are affected by burnout syndrome is that which are constantly under pressure, like doctors and nurses and teachers are the major target of the burnout syndrome. Now, what are the causes of burnout syndrome? There are many characteristics that affect to cause burnout syndrome. The job related problems are the first. They are not feeling unhealthy relationship with their colleagues or high workload. Unsatisfied backgrounds are so many reasons. Job characteristics that reason for burnout syndrome. Now family background is also a reason, can be also a reason for the job burnout. That is responsibilities and family pressures. So many people and many female mostly, they have to handle their work from house, their job, and they may find with burnout syndrome. Now, what are the signs and symptoms that are found during people during burnout syndromes are poor performance in their works, lack of concentration in their work, and take themselves isolate, increase accidents and mistakes, and cynicism, irritability, and constant depression, and stress, anxiety, forgetfulness, frequent illness, difficulty in sleeping. We can say that insomnia. And due to mental unhealthiness, people are going to suffer after some time in body. Body unhealthiness, that are blood pressure, heart related problems, hypertension, insomnia, etc. And how to handle burnout syndrome? So burnout syndrome is not medically diagnosed, and there is no diagnosis are there for burnout syndrome, but some are tricks and techniques from that people can handle their burnout syndrome. They can evaluate their job options. They can go for their satisfied job, seek support of any person, try to relaxing activity like meditation, get some exercise, get some sleep, and mindfulness are the handling of job burnout. Now the conclusion of after reading so many papers, research paper, I give a review on the burnout syndrome, and I conclude that according to the previous study reveals that burnout in professionals is a major growing psychological hazard due to many dissatisfied situations around the person. Study to a greater extent in job burnout is necessary, and a few papers, previous research papers are my references. Thank you.
Madam, you have written review. How many papers you have read for writing this review? Madam? Purvi,
Yes, ma'am. Yes, ma'am.
how many papers you have read for writing this review?
Ma'am, I have read around 7 to 8 papers of burnout syndrome on doctors, nurses, and I have read one in like IT companies, etc. Burnout syndrome surveys.
Okay. So, from which journal you have read that research papers? Tell me the name of few journals.
I don't remember. I remember a single journal, that is European Journal of that is written in 2017, I think.
2017.
Yes, ma'am.
Okay, but writing a review, it is a skilled job, and it require a lot of information and your research in that field, or you should know how many books are written on that. So keep in mind this.
Yes, ma'am.
Okay. Okay. Thank
Yes,
you.
ma'am. Thank you, ma'am.
Okay, thank you, Purvi. The next is PZ 17.
Madam, what is the 14, 15, 16?
PZ 13 has completed. Now the PZ 14, sorry, PZ 14. Yes.
Yes, ma'am.
Tangre PB, yes, please start your presentation. Sorry, sir.
कुठला PGZ
14.
14. Taxonomical study of fresh water. तीनच काही दिसत नाही.
मॅडम हे मला वाटतं
हा ओके ओके आता ठीक आहे. ओके 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 करेक्ट. हा
Okay. Good afternoon, everyone. I am Poonam Balasur Dangre, a student of BSc third year. I am studying in Mahadari Ecological Research Institute, and here in abstract, Mahadari is diverse region of pond, lake, and streams. This water bodies carries various freshwater fish species. A study was carried out on the diversity of fishes as well as its taxonomic classification. So, starting with introduction of my topic, Mahadari is very small forest covered area with vast biodiversity. Fish fauna of this small area is also diverse. As we know, fish is important component of food chain, hence it acts as a keystone species and indicator of healthy aquatic ecosystem. With these, fish also has nutritional values, medicinal, economical as well as aesthetic values in human betterment. The observation of fish was done from August 2021 till January 2022. And fishes which are caught by a local fisherman and were photographed and then identified by using keys of Jairam and the keys of Talwar and Jingran. The photographs of other fishes was taken from fish-based website. The study was done with the help of my mentor. Yes, ma'am.
No.
Continue.
All these photographs are your own.
Some photographs are my own, but some are we not able to capture photographs, so we collect it from internet.
No, how you are going to be matching to your identified and this internet photographs?
Sir, I'm not understanding. Sir, I'm not understanding.
How you are going to be identifying? How you are going to identify the similarities of that internet photographs and what you have observed these specimens?
Only some species of fishes. So we know about it, which type of fish, and we collect information from.
No, madam. Maximum photographs are internet. No.
No, ma'am. Maximum photos are.
See, how much you fish show here? तुम्ही
15
कुठलीही पाणीमध्ये नेट टाकलं तर हे सगळं मिळतं ना तुम्हाला.
हो सर.
मग तोच मध्ये काय नवीन काय आहे आता? आणि तो तुम्हाला नेटिंग माहिती आहे काय? हाऊ मेनी टाइप्स ऑफ नेट्स आर देयर फॉर फिशिंग?
No, sir. We collect from
नाही तेच म्हणतात आता काय आहे, आता तुम्ही कोण तर फिश काढणार आहे, कोण तर करणार आहे, तुम्ही ऑनलाईन मध्ये घेणार आहे. तुमचं एफर्ट काय आहे मला सांगा. व्हॉट युअर हॅव्हिंग युअर ओन एफर्ट.
Sir, we identify it and we conclude that 50 species of Cyprinidae family out of 22 species were observed in this area, and the dominant family is Cyprinidae family. So hence we want to conserve it. We don't introduce any other exotic species in that area.
Give me example of some of the exotic fish.
Sir, katla is an exotic fish, and
Already katla is there in your picture.
yes.
How you are going to be avoiding? Already that is in there, that river. What you are taking that pond?
Introduced from some
Okay.
तो कटला इंट्रोड्यूस केलेला आहे काही माणसांनी, त्याच्यामुळे तो तिथे आला असे. म्हणजे जास्त करून सायप्रेनिडी फॅमिली मधले आहेत, जे नेटिव्ह आहेत बऱ्यापैकी.
Okay, madam. Don't take the photograph from internet. Okay.
Okay, ma'am.
If you are claiming that we have collected, we have done, and previously anybody has done the biodiversity or the taxonomical study in that area, in that your water body, Mahadare.
Yes, my mentor. Yes.
Already. Who has done?
My mentor, Pradnya Mohite.
Pradnya Mohite has already done. And what you have done again? Or Pradnya Mohite has done and you are presenting what?
Taxonomic classification I done of this Cyprinidae family.
No, what I'm asking previously, other than you, whether anybody from your area or from some area, they have done the taxonomical study in that water body. They have collected. No, you are first time doing that one.
Yes, taxonomic classification was done by me only, I think. Yes.
You have not seen the review of literature. You have not seen the research paper.
मॅडम बीएससी स्टुडंट आहे मॅडम, काय कराल ते.
चला ठीक आहे, चला ओके.
Thank you, ma'am.
The next is PZ 15. Are you there? 4 CB pores. PZ 16 RJ Patil or SS Patil. Then PZ 17 Neha Desai. Yes, Neha Desai.
Yes, ma'am.
Please share your presentation. So this is PZ 17.
Okay.
Hello, good afternoon all. Myself Sneha Desai. Here
Hello,
I am
Sneha.
presenting my research work entitled.
Hello, Sneha.
Screen.
Yes, ma'am.
Screen
is not clear.
Okay.
And please hold it in horizontal way so that entire poster can be visible clearly.
मॅडम.
Hi, sir.
दीपाली मैडम थोड़ा मैडम ने सांगा कि कैसा पकड़ायचं ते.
Yes, sir. Sneha Desai, madam. Please hold your mobile in horizontal mode so that the poster
Okay.
is clearly visible.
Yes. Okay.
Good afternoon all. Myself Sneha Desai. Here I am presenting my research work entitled GCMS Analysis of Bioactive Components from the Seeds of Erythradia cardamom, a potential therapeutic plant, under the guidance of Dr. M.V. Vavekar. As we know, the medicinal plants are directly and indirectly used in the treatment of the various diseases, and also it is used to promote the general health. Phytochemicals play an important role in the drug formulation. The analysis and the extraction of the plant material like the leaves, seeds, stems are the most important because it play key role in the development of the herbal drug formulation. Hence, the present study was undertaken to find out the bioactive components present in the seeds extracts of the Electridia cardamom by the GCMS analysis. For this, we used the ethanol as a solvent and the phytochemical screening and GCMS analysis was done. In that, we observed that the extract contains the many bioactive important compounds like flavonoids, glycosides, terpenoids, steroids, and the saponins. The GCMS
ओके
analysis.
देसाई
हा सर.
तुम्ही काय काय आता प्लांट केमिकल सांगितलं ना, मला फक्त एकच सांगायचं, प्लांटचा केमिकल तो कुठल्या सॉल्व्हेंटमध्ये एक्स्ट्रॅक्ट होता?
ओके, इथेनॉलिक एक्स्ट्रॅक्ट होते सर.
इथेनॉल युजली ओनली इथेनॉल.
Ethanol, only ethanol it is used as the solvent.
Yeah.
And the seed powder.
Seed powder, okay, because according to this polarity of this organic solvent, they are able to separate the different components. What you said that alkaloids, tannins, terpenoids, like that.
Yes sir.
Ethanol मध्ये कुठली ऍक्टिव्ह कंपाऊंड बाहेर येतात?
Ethanol मध्ये mainly oily content and the organic molecules sir.
तसं सांगू नका. मग तो ऑर्गॅनिक आयल येणारच आहे. इट्स अ नॉन फॅट आणि फॅट येणार आहे. तोच मी ऍक्टिव्ह कंपोनेंट्स व्हॉट आय एम सेइंग यू व्हॉट इज ऍक्टिव्ह कंपोनेंट व्हॉट यू नीड. बिकॉज तो सीड मध्ये
फॉर दी बायोलॉजिकल ऍक्टिव्हिटी पर्पज सर. स्टेरॉइड्स, अल्कलॉइड्स सर.
स्टेरॉइड्स कुठली सॉल्व्हेंट मध्ये बाहेर येतात? अल्कलॉइड्स कुठली मध्ये बाहेर येतात?
Mainly ethanol, chloroform, hexane.
Because when you want to make because तुम्ही PhD वगैरे सगळं झालेलं आहे. Because when you want to make, because तोच मध्ये काय एक ऍक्टिव्ह इन्ग्रेडियंट तुम्हाला आहे, that you want to take that one means you have to be very precise. Because always your peak should be 1:1. If it's a preliminary, I am agree. It's a basic work, okay, I am agree, because this GCMS graph is showing that so many peaks here.
Yes sir.
आणि तुमचे एक्सट्रॅक्शन प्रोसिजर काय आहे?
सॉक्सेट एक्सट्रॅक्शन.
सॉक्सेट मध्ये आता कमी झाला, ओके, सॉक्सेट केलेलं आहे. इट्स ओके. मग आता ये GCMS सीड कार्डमम मध्ये ते हे काय वेलदोडा काय?
इलायची सर.
ये मॅडम इतनी कॉस्टली वेलदोडाला तुम्ही काय काढले तर तुम्हाला प्रॉफिटचं व्हॉट इज द कॉस्ट बेनिफिट रेशियो ऑफ दिस?
Sir, it is a small part of my research work actually.
नाही, because काय आहे, because if you are spending the 1,000 rupees, if you are getting the 5 rupees outcome means, because basically it is one of the spicy and it's very costliest biological sample, no?
Yes sir, but the compounds which are observed, for example alpha terpinyl acetate which is observed, it is highest percentage in this extract and it has the maximum role in the anti-cancer property and the neuroprotective effect. So in drug formulation sir, it is used.
What you are saying, I am 100% agree, but what I am advising you, it has become costly because basic thing is costly.
Yes sir.
त्यासाठी इसमें असलेलं काय केमिकल तुम्हाला सापडलेलं ना, तो केमिकल दुसऱ्या कुठली स्वस्तामध्ये आणि सोपं मध्ये मिळायचं प्लांट मध्ये मिळता का ते विचार करा पहिला.
Okay sir.
Because that's going to be a cost benefit ratio is there.
Okay, yes sir.
Is this extract you are used for any further use?
Yes ma'am, this extract it is used for the study of the anti-diabetic effect and it shows its effect, anti-diabetic effect in the mice.
Which active component it is acting as an anti-diabetic?
Actually ma'am, this is the crude extract and it consisting of the number of active compounds Okay, and these many other molecules in this present extract, they act synergistically. I'm not isolating the individual compounds. So the compounds which are present, so we conclude that the whole extract, it is considered as the anti-diabetic one, or it is controls the blood sugar level.
Okay, okay. So synergistic effect is there. Okay.
आणि
Synergistic effect.
आणि तुम्ही स्नेह आता परत तुम्ही प्रेझेन्स ऑफ फ्लॅव्हनॉइड्स अल्कलाइड्स वगैरे पॉझिटिव्ह केल्या प्रेझेन्स आहे म्हणून तो साठी इंडिव्हिज्युअल टेस्ट केला कायस का
हाँ सर, फाइटोकेमिकल स्क्रीनिंग, पॉझिटिव्ह निगेटिव्ह, ओनली कलर चेंज, कलर डिटेक्शन सर. प्रायमरी स्क्रीनिंग केली आणि फक्त त्याचे GCMS केलेले आहेत आपण.
ओके 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 ओके
हाँ सर.
ओके थँक्यू.
Okay.
Okay, thank you sir.
Thank you, Sneha ma'am.
Thank you.
Now the next is PZ18.
Yes, ma'am.
Suhas Dayal.
Can I start, ma'am?
Yes, yes.
Madam, 18.
Yes, sir. PZ18.
Okay. Suhas there.
Wait a minute, sir. I'm sharing my screen.
Okay, madam.
Wait a minute, sir.
Yes, we are waiting.
My screen is visible, sir.
No.
Hello, Suhas Dayal. Are you sharing?
Yes, yes, ma'am. Wait a minute. Just a minute, ma'am.
Yes, yes,
Ma'am, my
okay,
screen
okay.
is visible.
Yes, yes, start.
Hello, everyone. My name is Suhasudhir Dayal and my partner name is Namrata Rajendra Farande. So we are a student of MSc and department from a department of zoology, YCIS Satara. And our work name is effect of some medicinal herbs on a development of Bombax mori L, that is a mulberry silkworm. So sericulture is a rearing of a silkworm for the production of a natural fiber silk. In a sericulture, there are several commercial species of a silkworm, but Bombax mori L, it is a most widely and intensively studied. So silkworm is a larva of a caterpillar of a domestic silk moth, that is a Bombax mori. It is a typically monophagous insect and a mulberry leaf is its a sole food. In a recent years, many attempts have been made to improve the quality and the quantity of a silk through enhancing the leaves, which is by nutriting, spraying with antibiotics, vitamins, plant products, or using an extract of a plant. So the aim of our the aim of our work is to evaluate the effect of some medicinal herbs, that is a Mangista and Triphala, on a growth and development of a Bombax mori L. So basically, the Mangista and Triphala are herbs with a greater medicinal values used in Ayurveda. The Mangista is benefited for the best blood purifying herbs. It is a flowering plant, belongs to a coffee family, that is Rubiaceae, and the Triphala is an Ayurvedic Rasayana formula consisting of equal parts of three fruits, that is Amalika, Bibitaki, and the Haritaki. So this work
So our work is conducted in our laboratory of a Department of Zoology, YCIS Satara, as per the rearing methods described by Krishnan Swami, to know the effect of some medicinal herb on the development of Bombax moris. So basically, powder of a Mangista and Triphala was purchased in a local herbal medicinal store, and initially these both powders were separately dissolved in a distilled water through a 1% of a concentration. Okay, so healthy and uninjured third instar larva were selected for an experiment, and they were divided into 3 batches, that is control, treatment 1 and a treatment 2. So for a further experiment, for a further experiment where we for a control we were feed with the untreated fresh mulberry leaves, and wait a minute, and for a control, we were fed with the fresh mulberry leaves, and for a treated first batch were fed with the mangista, and a treated 2 batch was fed with a Triphala extract. So the freshly harvested mulberry leaves were dipped in the extract of a mangista and the Triphala. The mulberry leaves coated with the mangista and Triphala extract were air dried before providing to a treatment larva. Okay, so feeding with a treated larva to treated batch, and as well as a fresh mulberry leaves to a control batch, was conducted 3 times per day. During this experiment, the observation was recorded on the morphological characteristic.
सुहास.
Okay, sir, wait a minute, थोडंसं एक्सप्लेन करायचं मला अजून.
No, sir is asking. Listen.
नाही नाही, तुम्ही एक्सप्लेन काय करणार आहे? तुम्ही एक्सप्लेन करायच्या पेक्षा मला आन्सर दे ना बाई.
ओके सर.
हा, आता तुम्ही वापरलं 2 किडा आहेत का?
दोन नाही सर, वी आर युजिंग Bombax mori larva.
फोटोग्राफ मध्ये 2
yes sir, this one is treated, wait a minute sir, this one is treated and this is a control batch sir.
हो, तेच म्हणाले आता मी काय म्हणाले, 2 लार्वा दाखवले आहेत.
हा, येस सर.
ओके, हा, आणि ट्रीटेड मोठा आहे आता.
हा, येस सर.
आणि कंट्रोल बारीक आहे आता.
येस सर.
हा, आणि
बिकॉज ऑफ
नाही बिकॉज कुठं आहे, बिकॉज ते पुढचं आहे अजून. आता हे काळा का झालंय आता? Why it become black? Why this treated larva is black color?
Treatment, sir, because of treatment.
Because of treatment?
Yes, sir.
No, if that color is like that, it is not going to be survive, no? Because silkworm is a very sensitive one.
No, sir, it is a survive. Sir, I can't hear your question, sir.
Huh?
I can't understand your question actually.
Where you get the eggs? Where you get the eggs of this silkworm?
Sir, can you repeat your question? I can't understand.
सिल्कॉर्मचे अंडे कुठं आणलेले आहे?
Local sericulture farm, sir.
हा, व्हरायटी कोणचा आहे?
And we are variety is Bombax moriel.
Bombax moriel is scientific name is silkworm. Right now there is a different varieties are there. Mulberry silkworm. Right now there are so many breeds are there, silkworm breeds. Which breed you have used?
Bombax mori, sir. Bombax moriel.
By Bombax mori is the scientific name of the silkworm. Okay, तेच मध्ये ते काय म्हणतात मराठीमध्ये?
Sir, Bombax moriel is a scientific name and we are taken mulberry silkworm, sir.
Okay, तोच मध्ये कुठली जातीचे घेतलेले? There is so many varieties are there in silkworm.
Sir, the only I know is that is a mulberry silkworm, sir.
Okay, what you are saying that's a I am agree. What you are saying that's a mulberry silkworm.
Yes, sir.
आता ज्वारी तुम्हाला सिंपल में सांगतो, आमचं ज्वारी आहे ना जब पागरे करतात तो ज्वारी is having so many varieties like that. Silkworm is also so many varieties. कोण होतं तुमचं गाईड कोण होतं? Who is your guide?
जाधव.
गणेश जाधव.
Yes, sir.
Okay, good. पुढं पुढे काय केलं, काय cocoon weight वगैरे?
Yes, sir.
What is rendita?
These parameters we are calculating, sir.
What is rendita?
So our result shows that yes, sir. So our result shows that the extract of Mangista and Triphala has been found to be effective over the control, sir. So as shown in this table, the larval weight, larval height, cocoon height, cocoon width, and shell weight, pupal weight, and silk gland weight, these parameters are increased in a treatment batch as compared to control batch.
Okay. So very good work. How you measure the larval height?
By vernier caliber, sir.
Weight?
Hello, sir.
Okay. Weight?
By electronic balance.
What is the accuracy? What is the accuracy of electron balance?
What do you mean by, sir?
What is the accuracy of your electron balance? Okay, good. Very good.
Answer.
For a conclusion.
And our conclusion of our project is the growth and development of a larva and subsequent cocoon production is greatly influenced by our medicinal herbs. So Triphala and Mangista has a growth promoting effect on a silkworm, which
उठावीने
the commercial quality of silk and can be used in sericulture for a yield production, sir.
And how much quantity you have used for on that leaf? Whether you have measured that one, or just you have put something amount of something that Triphala, some amount of that.
No, ma'am. Hello, ma'am.
Calculated.
Yes, ma'am. We are calculated. Actually, in control, we are taken 3 batches, ma'am. Like control treatment.
Just I am asking.
1% concentration.
But whether that your larvae has feed all the plant, all the leaf, you have studied that also? How much have taken that?
Yes, ma'am. We are feeding our larva 3 times per day, ma'am.
Okay, and these photographs you have taken your own or from it is internet?
No, ma'am. It's my own photographs.
All these photographs are you have taken from your camera?
Yes,
you have
ma'am.
taken from your camera? Okay,
definitely, ma'am. Yes.
Okay.
And it clearly shows that, ma'am, all morphological characters are greatly influenced by our herbs, ma'am.
Okay.
Treatment. Okay, ma'am. Can I stop, ma'am?
Yes. Thank you.
Yes. Good work.
Thank you, ma'am. Thank you, sir. Thank you.
Okay, the next number is PZ19. Amruta Nangre.
Yes, ma'am.
Yes, please.
Yes, ma'am.
Yes.
Yes. Ma'am, screen is visible?
Yes.
Good evening, ma'am. Good evening, sir. I am going to present a poster on the biodiversity conservation. What is the why biodiversity should be conserved, or
Madam,
why should
just तुमचं थोडं ओळख करून दे मॅडम. Just introduce yourself.
My name is Amruta Nangre from Rajaram Bapu College of Pharmacy, Kasegaon.
You are studying
B Farm.
B Farm.
Yes,
what is the relation between your pharmacology, pharmacy and biodiversity?
Not actually, sir, but I want to participate in this competition, so I I selected
Okay,
this.
you participated, you are getting the certificate, don't worry. That things is okay. Whatever having that interest, no, that is going to be fulfilled. But what is the relation between your pharmacy and this biodiversity conservation?
No, not actually, sir.
Huh? There is no any link, no.
No, sir. Actually, biodiversity is related with every field. It is a life science, so I can also participate it.
No, you can participate. Oh, madam, I never say don't participate. You have to participate. But how you are going to be correlating your pharmacy and this biodiversity conservation? And have you conducted any experiments in biodiversity conservation? Because this is showing you some of the photo, that's all.
No, sir. I have not conducted, but I have studied.
What you have studied? You have just read. Yes, you have just read papers and you have collected information. That is what.
No, madam, this all the things is a PPT or this all the information is downloaded from the internet. That's all, nothing more than that in this PPT.
Yes, sir.
Okay, what is biodiversity?
Biodiversity is totality of genes, species, and ecosystem of the region.
Because Indian peoples are doing more biodiversity work rather than the European peoples. Why?
Sir, I didn't got your question.
भारतमध्ये किती लोक बायोडायव्हर्सिटी अभ्यास करताना त्यापेक्षा कमी अभ्यास युरोपमध्ये करता काय करता बायोडायव्हर्सिटी युरोपमध्ये कमी करता भारतमध्ये ज्यादा करता हे बायोडायव्हर्सिटी
because biodiversity because Indians are trying to conserve biodiversity by various ways such as in India there are 104 national parks are and 18 biosphere reserves. Then wildlife sanctuaries are also therein. There are 551 wildlife sanctuaries in India.
Madam, what is endangered species? Tell me the name of your endangered species.
Endangered species means the species which are now are going to extinct. In which blue whale, Asian elephants are there, and tiger species are there.
What is the red book?
Red book, it is book which is in which endangered species are registered.
Okay. Because you were having the interest. You were having the interest in biodiversity, but field is a study field is a different one. It's okay. Let it be. Good. Very good.
Okay.
Thank you, sir.
Okay.
Thank you.
The next is PZ 20.
Yes, ma'am. Can I audible?
Yes. Please start, Shrutika.
Mr. Wait, ma'am. I'll share my screen.
Okay.
Screen is visible, ma'am.
Please hold your mobile in horizontal way.
Okay. Just a minute, ma'am.
Yes. Now it is okay.
Okay. Can I start, ma'am?
Yes.
Good
Madam,
evening, all.
Just
yes,
wait.
ma'am.
You have collected all these photographs from internet?
No, ma'am. Just pictures clicked by me or some from internet, like antenna, frenulum, and eyes.
Which picture you have taken as a photograph and which from internet? Just tell me which photograph you have taken from your camera.
Moth, butterfly, body covering
Ah.
of moth, and food collection in moths. Or food covering both insects. Sorry, body covering, ma'am. Sorry.
Body covering.
Huh.
And sitting position moth. Only 2 pictures
Yes,
you have taken.
yes. No, ma'am. Sitting position of moth and butterfly, body covering of moth and butterfly, and food collection of moths.
This food collection.
Spinged moth.
Hello.
This is okay. This is a spinged moth which is collecting the food. Yes.
Food collection. Which is that moth?
Don't have any idea.
You have taken that photograph.
Yes,
and
ma'am.
you
Yes.
don't know. So just you have taken photograph. It is not necessary to find out which is that moth or which is that butterfly. You should know the name of that. Otherwise, if you don't know the name, then why you are presenting here?
Sorry, ma'am.
What is the function of this feathery antennae in moths?
Sir.
What is the function of this moth's antennae, feathery antennae?
Moths are pollinators, sir. Lepidoptera contain around 86% of moths and 14% of butterflies, but maximum pollinators from moths.
Okay, what is the function of this antenna? Moth मध्ये असलेल्या antenna काय काम आहे?
Okay, sir. Moths and butterflies are use their antennas to sense the things in the environment, like finding other insects and feeling around the plants.
Hello.
Okay.
Can I start, sir?
Start. Okay. What you are going to be starting?
Hello.
Start, kara.
Okay. Thank you, sir. Good evening, all. My name is Shrutika Rasa. I'm here to present my poster on a difference between a moth and butterfly. Moths and butterflies are almost look same, but people do not understand the difference between a moth and butterfly. There are 2 divisions of the order Lepidoptera, namely Heterocera means moths and Rhopalocera means butterflies. So we studied these organisms. Some organisms are have been noticed during the daytime, or other have been spotted during the night time. These insects were observed in 4 life stages, including egg, caterpillar, pupa, and adult. Transformation from caterpillar to pupa is one of the actual wonders of nature. So as per our observation, moths are nocturnal and butterflies are diurnal. There are some exceptions in the both the insects. If you look attentively, you can easily identify the moth and butterfly by their sitting position. Moths keep their wings horizontal on back while resting, and butterflies are keep their wings vertical on back while resting. Next character is body covering. Moths have a hair-clad body and butterflies have a smooth and lean body. Now moving to the next point, which is morphological character, which is eyes. Moths have a superposition eyes. This type of eyes create image up to 1000% brighter, but sacrifice with the quality of image. And other hand, butterflies have a opposition eyes, which work by combining the multiple images. Another character is food collection. Moth collect their food during a night time, and butterflies are collect their food during a daytime. Next differentiating character is frenulum. Moths have a frenulum, which is wing coupling device, and this device used during flight. Butterflies do not have a frenulums. Next character is antenna. As I say, moth and butterflies are use their antenna to sense the things in the environment and feeling around the plants. Butterflies have 4 different types of antennas, such as filiform, fusiform, unipectinate, and bipectinate. Filiform is a basic form of antenna. Filiform antenna.
Okay, madam. Okay, thank you. And whenever you are taking any photograph from internet, you have to quote it that it is taken from internet. Okay, because if you want to publish, if you want to present, because I think most of the photographs, I think it is from internet.
Yes,
Even
ma'am.
body collection also you have not taken, because I can see it on internet now.
No, ma'am. Moth
Body
body covering is taken by me.
body
Because
food
Oh, sorry.
collection, sorry, food collection.
Yes.
Okay, so whenever you are presenting, whenever you're publishing, keep in mind that if you are taking the photograph from internet, you have to quote it, that is, I have taken it, or from which source. Otherwise it become as a plagiarism. Okay, thank you, madam.
Okay, thank you, Shrutika. Then the next is PZ21.
And so
PZ21. I think the group members are not here, ma'am. We will continue.
next, 22.
PZ22.
जे झाले त्यांना रिमूव्ह करा, म्हणजे लक्षात येईल आपल्याला किती राहिले ते.
मी मगाशीपासून तेच
Actually
म्हणालो,
तेच
जरा
सांगते
मार्क
ती.
काढायचा.
Yes, sir. I
म्हणजे
removed
लक्षात
them.
येते हो, कारण सगळेच अजून इथे दिसायला लागलेत, काय कळेच ना किती राहिले.
I removed some participants, but they are requesting that they want to listen other words. So again, they
Okay, बरं ठीक
No,
आहे.
no. Because online only examiners were examiners. That's all.
Okay, sir. Okay.
तर लक्षात येत नाही किती राहिले तो.
हा बरोबर.
नाही किती राहिले, आणि दुसरं त्यांचं म्यूट नसतं, बॅकग्राऊंड म्युझिक आवाज येत असता.
Yes.
तर नेक्स्ट कोण? 23, 24, 25 कोण राहिले का?
25 madam. I am.
हा. Omkar, please start.
Okay, start.
What is number?
25.
Yes, ma'am.
Yes.
Start.
मैडम नंबर काय आहे इसका?
25.
Mimicry of snakes.
Good afternoon, sir. My name is
Omkar,
Omkar Ale.
hello. Omkar, please hold your mobile in horizontal view.
This time, madam.
नाही.
आडवा पकड.
आडवा करा, आडवा.
एकच 1 minute.
मोबाईल आडवा करा की.
नाही त्यांनी स्क्रीनच लॉक केली आहे का काय बहुतेक, नाही दिसतच नाहीये काय. हा ठीक आहे, दिसलं. ओके. हा ठीक आहे बा.
Good afternoon, all of. My name is Omkar Ale. I am snake rescuer and also art student. My topic for poster presentation in mimicry in snakes. I am going to present my poster in Marathi. साप हा सरपटणारा प्राणी आहे. काही विषारी आणि बिनविषारी साप हे स्वतःचे संरक्षण करण्यासाठी एकमेकांची नक्कल करतात. मात्र या सापांची माहिती नसल्याने लोक बिनविषारी सापांना विषारी साप समजतात आणि त्यांना मारतात. हे साप हे शेतकऱ्यांचे सर्वात चांगले मित्र आहेत, कारण ते शेतात बायो कंट्रोल एजंट म्हणून काम करतात. परिणामी अशा सापांचे संरक्षण करण्यासाठी जनजागृती व्हायला हवी. सातारा शहरात 50 किलोमीटर परिसरात हे साप मी पाहिले आहेत.
हे तुम्ही स्वतः काढलेले फोटो आहेत काय हो?
हो सर, हो मॅडम.
काय इंटरनेट वरून घेतलेले आहेत?
नाही, मी काढलेले, स्वतः फोटो मी काढलेले.
आतापर्यंत किती सापांना रेस्क्यू केलं तुम्ही?
आतापर्यंत असे 3,000 ते 3,500 साप.
कधीपासून करता हे काम?
हे मी 2018 म्हणजे माझा 18 कम्प्लीट झाल्यापासून मी ट्रेन केलं, त्यानंतर कम्प्लीट केलं.
कुठे कोर्स केलाय का तुम्ही?
हा,
कोर्स केलाय का कुठं?
ह्याचं ट्रेनिंग घेतलं मॅडम. स्नेक रेस्क्यूअर आहे मी. ह्याचं ट्रेनिंग घेतलं, साप कशी पकडायची काय.
ओके, युअर टायटल इज मिमिक्री इन स्नेक्स. काय सांगा, व्हॉट इज मिमिक्री?
मिमिक्री म्हणजे साप नक्कल करणे, एकमेकाची नक्कल करणे.
म्हणजे काय करतात ते?
बिनविषारी साप हे स्वतःचे बचाव करण्यासाठी विषारी सापांचा स्वतःचं संरक्षण करण्यासाठी विषारी सापांसारखे दिसतात, त्यांची नक्कल करतात, जेणेकरून त्यांना कोणी मारायला नको.
कोणचं स्नेक ऐसा करता सांगा की, नाव सांगा की.
जसं की आता आपल्या फोटोमध्ये इथे दिसतो तो नाग आहे, पहिला आहे तो भारतीय नाग. त्याची नक्कल करणारा साप आहे तो पट्टेरी धुळनागिन. हे दोन्ही साप एकसारखे दिसतात. नागाला ह्या दोन्ही सापांना ओळखायचे कसे, तर नाग हा फणा काढतो व धुळनागिन हे धुळनागिनचं तोंड हे निमुळतं असतं. त्याच्यानंतर नक्कल दुसरा साप आहे तो चापड्या, तो विषारी साप आहे. आणि दुसरा आहे तो गवत्या, बिनविषारी साप आहे. हे दोन्ही साप एकसारखे एकमेकांची नक्कल करतात. जसं ह्या दोघां सापांना ओळखायचं असेल तर चापड्या हा जर तोंड निमुळत, मान त्याची निमुळती असती, चपटी, व गवत्या हा प्लेन एका ह्याच्यात कलर मध्ये असतो. तिसरा येणारा साप आहे फुरसे, त्याची नक्कल करणारा आहे साप मांजऱ्या. फुरसे हा विषारी साप आहे आणि मांजऱ्या हा निमविषारी साप आहे. मांजऱ्या हा साप झाडावर असतो आणि फुरसे हा दगडांच्या विटांमध्ये हा सापडणारा साप आहे. ह्या दोन्ही सापांना ओळखायचे कसे, तर फुरसे हा लांबीला छोटा येतो, जाडीला मोठा येतो. आणि मांजऱ्या हा साप लांबीला मोठा येतो आणि पातळ येतो. त्यांना ओळखायचे कसे, तर त्यांच्या डोक्यावर, फुरशीच्या डोक्यावर बाण आकृती असतो, धनुष्यबाण. आणि मांजऱ्या सापाच्या डोक्यावर इंग्लिश मधला वाय आकृती त्या टाइप मध्ये असतो. त्यानंतर येतो
तुम्हाला
घोणस.
अजगर पण दिसला काय होती ते? अजगर पण दिसला?
हो, आहे की.
अजगर कुठे आहे मॅडम?
तिथे लिहिलंय
आहे
की
की,
खाली
आहे.
अजगर, ते बघा.
आहे.
बापरे.
घोणस, त्याच्या खाली आहे अजगर.
अजगर
घोणस
हे अजगर म्हणजे काय म्हणता मॅडम?
पायथॉन, पायथॉन, पायथॉन.
इंडियन रॉक
पायथॉन इथं आमच्या इंडियामध्ये कुठे आहे हो मॅडम?
तिथं लिहिलंय की पायथॉन आहे.
हे कुठे आहे? पायथॉन नाही
सापडलाय
इथं.
ना तुम्हाला तिथे? सापडलाय का अजगर तुम्हाला इकडे?
हो, सापडलाय.
आहे म्हणतोय की.
हे सगळे साप मी पाहिलेले, स्वतः डोळ्याने पाहिलेले साप आहेत मॅडम. मी फोटो
आणि
काढले.
आयडेंटिफिकेशन तुम्ही स्वतःच करता काय?
हो, मी ते आपले नीलकुमार खैरे सर, त्यांच्या पुस्तकात बघून मी आयडेंटिफिकेशन करत गेलो. त्याच्या पुढे त्याचं ते शिकलो व्यवस्थित. त्याचं पुस्तकात बघून मी आयडेंटिफिकेशन केली त्याची.
बर, ठीक आहे. ओके.
वरती असलेला कुठले रे? एक कुठे, दोन साइड मध्ये, राईट लास्ट तीनो.
लास्ट मन्यार आहे.
लास्ट वन मन्यार सर. लास्ट आहे ना से मन्यार आहे, मन्यार.
आणि त्याच्या खाली येणारा तो कावड्या, त्याची नक्कल करणारा. तर कंटिन्यू करू सर?
ठीक आहे ना. ओके, बस.
ठीक आहे.
ओके, थँक्यू.
ओके, थँक्यू.
थँक्यू ओंकार. Is the Namrata Dangi there? Namrata Dangi. Nisha
कोणाचं राहिलंय काय फक्त विचारा आता.
yes.
नसेल तर हे असं.
If the participants whose names were not announced here, and if they are having their names in the poster list, they can present. Or please tell us.
येस हे सगळ्यांचं झालंय ना मॅडम? वर नावं आहेत बघा ती सगळी.
Yes ma'am, yes.
दीपाली
Yes.
जरा काढून टाका हो मॅडम, ते काढून
वर्ष
टाकलं
नाही
मॅडम,
झालं,
लक्षात येतं.
संपलं सर.
Yes.
संपलं सर.
संपलं ना?
संपलं. हा, चला. आपल्यालाच काढून टाकणार आहेत आता ते.
No. On the behalf of my college, I would like to thank you both of you with my heart because you gave us your valuable time. You spend us your valuable time with us, and thank you for examining this session. Thank you one and all. Thank you all the participants. And soon, ma'am and sir will share the numbers from these presented posters, and that names will be declared in tomorrow's valedictory session. Thank you once again, one and all. Bye and have a nice day ahead.
Okay, thank you, madam.
Sir, thank you.
Thank you.
With the permission of examiners, I will conclude this session and I will end from my side. Thank you.
Okay, thank you, ma'am.